All right, we are now on the fourth module, and the topic that we have is called single factor and multi-factor experiments. All right, so this would be very quick, and we're not going to have some calculations because actually this is just an application of the topics that we had from modules one up to three. Um, and this will be better, or you can appreciate this topic better once we have our modules 5 to 8 also, wherein we are going to make use of some computerized tools or digital tools like Microsoft Excel and Python programming to conduct some statistical and data analysis procedures. Right? So let's proceed. Right. Before we talk about experiments, I have to emphasize this one, although we already had discussion on this during module one. All right. So just a recap, we have engineering design process. This is used in engineering to identify a problem, develop potential solutions, test prototypes, and refine designs, often involving experiments to optimize performance. So from here up to here, the end product is an engineering design, normally in the form of a prototype. Okay. Whereas in the scientific method, we are referring to a systematic approach to research, to answer questions or to solve problems. Right. So we have a question: why is the moon so bright, or why the weather is so hot today? Why are there people getting sick at work, or why some students fail to get or achieve the minimum passing score. So we are asking questions, and we need to find an answer to those questions. So we can do that using the scientific method, right? And it involves also experimentation. And the experiment here, we are doing the experiment to test our hypothesis. And in most cases in engineering context, we experiment using the prototype that we had built. Okay, so usually we conduct this, come up with a prototype, and then proceed here. Okay, before we can proceed at this point of our scientific method. So we apply our prototype or our engineering design here, and then continue. Sometimes we are um, at this point, when we brainstorm, evaluate, and choose a solution during our engineering design process, we can only proceed, or we can only get through this point once we perform scientific method, because there are questions that might need an experimentation also, right? So either way, um, there is an experiment involved, and possibly both of these might be um, dependent with the other. Okay, so my point is, in engineering, we are normally involved in both of these things. Okay, scientific method and engineering design. All right, so experiments. We are talking about experiments. When we say experiments, these are controlled procedures carried out to test hypotheses or investigate relationships between variables. So, especially in the case of our prototyping design. And we do that by manipulating specific factors and observing outcomes. Right? For example, you are building a prototype, you try to adjust this thing and then observe what happens. Right? So that's actually a form of experiment already. Um, Also, experiments are operations or procedures carried out under controlled conditions. I have to emphasize the word controlled. So it has been mentioned in the first definition and also here. Controlled conditions in order to discover an unknown effect or law. So we have to make sure, when we experiment, we make sure that the environment is controlled as well as other factor sorry factors are controlled, right? And we do that to test or establish a hypothesis or to illustrate a known law, right? So experiment, for example, we have this. Okay, let me zoom in. Okay, when we experiment, we have an input, and then, of course, after the experiment, we come up with an output. Okay, so here are examples of factors, variables, inputs. So for example, we have categorical factors. Category could be either a yellow corn or a white corn. So two groups. And then we have the numerical factors such as the things that we measure, like the amount of water, the amount of fertilizer, the type of soil. Okay, so we have categorical and numerical. Remember, if we have these variables, um, okay, okay, or later. Um, and then when we experiment, we have to set our control factors or the control conditions. So like the fixed temperature, the duration, especially when using the microwave. Um, what else? Right, we control them. In we separate our experiment into two groups, one that remains with one setting only, and the other with variable settings. Okay, so what else? Expected output for this, of course, um, our outcome, our result, our output variable would be the popcorn made. Okay, so we're trying to check whether the plant. Okay, the things that we input in our plant has okay, I guess these are two separate experiments. Okay, the one is about popcorn and the other one is about growing a plant. Okay. Um for the first one, whether the setting in our microwave oven has a significant effect in our output popcorn. Okay. While the second one, whether our the measures of our ingredients for growing the plant will have an effect on the height of the plant. Okay. So um that's an overview of experiments. Okay. So when we do experiments, we are dealing with variables, which are quantities or properties that can change in an experiment, such as the temperature, the pressure, and so on. Okay, when we say factors, we are referring to the specific independent variables that are deliberately manipulated to study their effect on responses. So, meaning variables can be an independent or a dependent variable, such as for this example, in this experiment, our independent variable is caffeine. Okay, so um, and that caffeine is being tested whether it influences our dependent variable, which is the number of words recalled. So to do this, we are questioning caffeine as a factor. Is it a factor for the number of words recalled? Okay, so to do that, we divide our respondents. We should have respondents, ideally human beings. But if we do that, make sure we have we do the ethical process. Um, okay, and we perform random assignment. Randomly, we assign some of them or half of them in our experimental group, while the other half in our control group. In the control group, we don't have any variations, or I mean we don't do any adjustments. We just secure a fixed setting of our conditions like temperature, the the number of things being consumed, or the amount of being consumed. What else? The duration of the experiment or each trial. So such things we just have them fixed, okay, throughout the experiment. While for the experimental group, we one of those factors are being manipulated. so that we'll see whether manipulating them will have an effect um, on the dependent variable that we are studying. Okay, so in experiments, scientists compare a control group and an experimental group that is identical in all respects. Okay, but during the experiment, one of them will be varied in terms of one or more factors. Right? So unlike the experimental group, the control group is not exposed to the variable under investigation or the factor being tested. And it provides a baseline against which any changes in the experimental group can be compared. Okay, so just consider the control group as our reference, and we'll see later if there is a significant difference between the two groups. Right? Now, here's a segue, side note, how to identify the dependent variable. So we can identify it by answering these questions. One is there, is this variable manipulated by researchers? So if yes, then that's an independent variable. But if we don't manipulate it, then that's the dependent variable. Okay. Or another question, does this variable cause an outcome? If yes, then that's an independent variable. If no, then that's dependent. Okay. And third possible question is this variable the result of another variable? So this time, if the answer is yes, then that's a dependent variable because the result is the dependent. And if no, of course, then that's the independent variable. Okay, so just determine which one causes which. Okay, which one affects the other. Right. So variables and factors. Remember, only the independent variables can be your factors. Okay, we can have one or more factors. All right, let's formally define the control and experiment or treatment group. Um, Control group refers to a baseline group in an experiment that does not receive the experimental treatment, meaning the manipulation, used for comparison to assess the effect of treatment. So remember, control group, no manipulation. Treatment group or experimental group refers to the group subjected to the experimental condition or treatment to measure its effect in comparison with the control group. So the process of aligning experimental units or participants to different groups randomly to minimize bias and ensure comparable groups. And it is called random assignment. So that is what I mentioned earlier. When we assign participants or um, respondents in either of the two groups, we should do it randomly. Okay, like for example, the rats. When we assign some of them in our control group or in our treatment group one, or actually we can have multiple treatment or experimental groups. Okay, but again, the point is we randomly assign them in these groups. Okay, so for example, subjects which are unknown values, if we do a random assignment, we can have control group and treatment group. Okay, for known values. All right. So we have an example here. Let's zoom in. Okay, we have these mice or rats in our groups, in different groups. Okay, um, our control group, or I mean, our control factor could be the housing condition. So everything, meaning these three groups have the same type of house, lighting in that house, temperature in that house, and other environmental conditions within the housing. Okay. Also, another control factor that we have is the diet. Okay, so same thing that we feed to the rats, not just the type of food, but also the amount and the manner in which we feed them. So the time, the duration, and what else? Everything else regarding the diet. Okay, the only thing that differs right now is the independent variable that we are studying. Okay. In the control group, we are giving them only the placebo. I'm not sure what this is. It's a certain kind of medicine for rats, I think. Okay. While in treatment group one, we give them instead a moderate dose of growth hormone. While on the second treatment group, we give them high dose of growth hormone. Okay. So we meaning for the two treatment groups, we give them a different medicine, and the first one has moderate only, while the second one has high dose. So now we compare them with that control group. You can compare them with each other, and eventually we'll see whether there is a significant effect of that independent variable, or that's medicine, and the dosage of that medicine to the um probably the behavior or the growth of the mice or the rats, right? So that could be our experiment. Okay. And of course, when you experiment, make sure you know in the first place why are you doing that experiment. Okay. So um a few what do you call this? Um hmm, uh Clarification. So we have the term experimental design and plan. When say experimental design, we are referring to the structured framework for conducting an experiment. And there we are specifying how treatments will be applied, what are the variables controlled, and what are the responses measured to ensure valid and reliable results. Okay. So on the other hand, when we say experimental plan, we are doing a detailed outline of how the experiment will be conducted. So experimental design, what's in our experiment? Experimental plan, how to do the experiment. Okay, because these two are sometimes being interchanged, but they actually have a slight difference with each other, right? So, for example, here we have an experiment, and we have this plan, whatever this plan is. The involved factors are the oven, the sugar, the flour, the eggs. Okay, so these are inputs, or maybe meaning are independent variable. So um our controlled variables are could be our limits, the setting of our temperature, the measurements of the water we add to the ingredients, or the number of eggs, number of um, amount of substance of whatever we add um, to our ingredients. Okay, so before we do that, make sure we have a plan. Okay, how do we do this? Um, okay, by doing a certain or preparing a certain methodology, right? So once you have ensured that you have a plan, then you conduct the experiment, okay? And then of course you will get the responses, right? So by the way, we have another example of control and experimental group here, right? So meaning it's not enough that we know what's in our experiment. Okay, we should also prepare um our plan on how to actually perform the experiment, right? Now this one I'm not going to discuss this in this case. These are um highly technical, right? Highly advanced for beginners in learning data analysis, right? But I'm going to introduce only the terms. You can take a screenshot of each. Okay, one of the most commonly used types of experiments is the completely randomized single factor experiments. Actually, the examples that we had um, um, earlier, plus the examples that I'm going to um, feature in a separate video, are actually uh, single factor experiments. Okay, so these are okay, I'm not going to single factor, meaning we are considering one factor only, right? And then we have the term blocking, and blocking involves grouping experimental units, subjects, samples into blocks based on known variables, sources, variability that are not of primary interest but can affect the response variables. Blocks help reduce variability within groups and improve the precision of estimates by accounting for sources of variation. So, let's consider a study testing the effects of different fertilizer, or that would be our factor A, and the irrigation levels. So another factor. So this time it's no longer single factor. We are considering two factors. So the blocks could represent different plots of land with varying soil characteristics. And by blocking, we ensure that each treatment combination is tested across different soil conditions, reducing the impact of soil variability on crop yield measurements. All right. Or in other words, we make sure that, for example, if there's an even um, quality or condition of soil in the area that we are going to experiment on, then we have to make sure that both groups have the same amount of good quality, moderate quality, and poor quality of soil. Okay, so we have to divide that part. So meaning if this portion is good, next is the bad, or the moderate, and the next one is the bad, then ideally we divide it this way, longitudinal. Okay, so that both groups can have a good soil, both groups can have moderate, and both groups can have a bad soil. Okay, so that means blocking. Okay, so in this case, the type of soil or the quality of soil is considered the blocking factor. Okay, so that would be for this one. We also have the confounding factors. Um, we mentioned something about confounding during the module 3 in the correlation part. So confounding occurs when the effects of multiple factors cannot be separated or distinguished from each other in the experimental design. Confounded effects lead to ambiguity in interpreting the results, as the observed differences may be due to the factors of interest or their interactions with confounding factors. Or in short, let's recall the ice cream and drowning incident example that we have in correlation. So we said there that um, it has been observed that whenever the ice cream sales go up, the number of drowning incidents also goes up. Okay, so there has been a positive, a direct correlation between the two, but those two are not actually connected with each other. Or I mean, physically, there's no physical formula that could connect the two. Okay, but there is a confounding factor that connects the two. Okay, so for example, the exposure and the outcome. So in this case, in our example, the ice cream and the drowning incident. So they're not really connected, but because of the confounding factor, they get connected. There has been a correlation between them. Okay, so I guess that would be my explanation about confounding. Right, so let's end with this one and let's continue. See you in the next part.